TY B.Sc ZY 332 Paper 2 Mammalian Histology Semester 1. Today we'll see chapter number 3, Histology Study of Different Organs. In that we'll see 3.3 topic, tongue. The tongue consists of freely movable portion called as body located in an oral cavity and a base or a root attached to the floor and forming part of an anterior wall of pharynx. On the upper or dorsal surface of tongue is a V-shaped groove called as sulcus terminalis with the apex of V shape directed posteriorly. This sulcus divides the tongue into anterior and posterior regions. The tongue is covered by a mucous membrane and its bulk consists of striated muscle fibers and glands. The muscle fibers are both intrinsic and extrinsic. That is, some are confined to the tongue whereas others originate outside, principally all on the mandibles and hyoid bones, and pass it into it. Between muscle fibers are glands. These glands are mainly mucous in the base of the tongue. Their ducts open behind the sulcus terminalis and the gland serous in the body of a tongue, their ducts opening anterior to the sulcus. And one more gland is mix acini, which is near the tip. Their ducts open on the interior surface of the tongue. The posterior third of the tongue has a nodular irregular surface because of a presence of lymphatic nodules. Between the bulging or cleft-like depressions of the surface epithelium termed as a crypts. Here the epithelium is infiltrated with numerous lymphocytes. The mucous membrane on the undersurface of tongue is smooth and underlined by submucosa. On the upper surface, the mucus shows numerous small protuberances called papillae, which gives the tongue a furred or ruffed appearance. There are 4 types of papillae present on a surface of tongue. First one is a filiform papillae, located over the entire tongue surface, mainly in rows parallel to the V-shaped sulcus. They are of a slender, conical shape and 2 to 3 mm in height, each with a primary core of connective tissues of lamella propria with secondary papillae. The covering epithelium is often partially keratinized and quite hard. Second papilla is a fungiform papillae. They are disposed singly among the rows of filiform papillae and are more numerous towards the top of the tongue. The shape is like a mushroom and a short stalk and a broader cap. The connective tissue shows secondary papillae and the covering epithelium is quite thin, so that the rich vascular plexus within the laminae propria impart a pink or reddish tinge to the papillae. Taste buds are found in the epithelium. Third papillae is a circumvallate papillae or vellum, like a wall. Number of these papillae, only 10 to 14 in man, and are located along the V-shaped sulcus. Each protuberance slightly formed from the surface and is surrounded by moat-like circular furrow with numerous taste buds in the epithelium of lateral wall, that is in the side of a circular furrow. Opening into the depth of the furrow are the ducts of serous Ebner's glands, which are located more deeply in the tongue. These thin serous secretions of these glands washes away food material from the furrows, permitting receptions of new gustatory stimuli by the taste bud. Fourth type of papillae is a foliate papillae. They appear as a leaf-like folds on the posterior lateral margin of tongue, with taste buds in the grooves between the folds. As in circumvallate papillae, serous glands drain into the depth of the groove. All papillae contain numerous sensory nerve endings for touch. Taste buds are associated with all except the piliform type. Now we'll see a taste buds. These taste buds containing gustatory, that is taste receptor cells. Taste buds lie in the oral stratified squamous epithelium, mainly in relation to papillae, but also elsewhere in the oral cavity, palate, and epiglottis. They are recognized easily on light microscopy as pale barrel-shaped bodies lying in the epithelium, extending from its basal lamellae through to the surface, but at the surface they are slightly depressed at the taste pore, a small apertures providing communications with the exterior. There are three cell types present in a taste bud: a supporting or subtentacular cells that lies mainly at the periphery of a taste bud, arranged like a stews of a barrel. Between them, more centrally, lie lighter staining, that is neuroepithelial taste cells. Usually there are 10 to 14 in each taste bud. Both types have long apical microvilli or taste hairs projecting into the taste pore. The microvilli lie in an amorphous polysaccharide material that is probably secreted by the subtentacular cells. The third type cell is a basal cell that lying peripherally near the basal lamella and is believed to be the stem cells for the other cell types. There is a relatively rapid turnover of cells with a lifespan in the order of 10 days, and it may be that the subtentacular cells is an intermediate state in the differentiation of sensory cells. Chemical stimuli are received by the sensory cells and transmitted by neurotransmitter to cup-shaped nerve ending lying between the cells as described here. Serous glands in relation to taste buds of valate and foliate papillae wash away food material and permit new taste stimuli, and similar serous glands act in a like manner in relation to taste buds located elsewhere. Only four fundamental taste sensations can be detected, and there is a regional sensitivity on the tongue. Sensation of sweet and salts are appreciated at the tip, whereas sour, that is acidic taste, at the side, and a bitter in the region of circumvallate papillae. Recently, it has been demonstrated that a single taste bud and a single papillae can respond to all four basic taste qualities. Certainly, no structural difference have been found to explain differences in sensitivity. Nerves from taste buds in the anterior two-thirds of tongue pass in the chorda tympani branch of a facial nerves. Those from the taste buds in the posterior third pass in the glossopharyngeal nerves, and those carrying taste sensations from the epiglottis and lower pharynx passes in the vagus nerve. All nerves lose their myelin before reaching to the taste buds. TY B.Sc. Zoology Mammalian Histology, chapter number 3, Histology study of different organs. In this we'll see 3.2 tooth histology of. The study of teeth is referred as odontology. Embryologically, the dentin, cementum pulp, and periodontal ligaments of teeth come from the mesoderm, whereas the enamel is a highly specialized ectoderm structure. Teeth-like bones are best studied in ground preparations or decalcified sections. Anatomical features of tooth. A crown is a protruding part above the gum level and is capped with enamel. Root is a part that is embedded 1 to 3 mm below the gum level and is covered by cementum. Neck is the junction of root crown. Alveolus is a socket in a jawbone that accommodates the tooth. Periodontal membrane anchors root to the alveolar wall. Apical foramen is a canal that opens at the apex of root and leads into the pulp cavity. And pulp cavity is a central cavity that extends from crown into the root canals and is filled with the dental pulp, a soft core of loosely arranged connective tissues. Now, dental tissue. A detailed histology of a simple one root tooth is described for simplicity. Here, each tooth is composed of two types of tissue, soft and hard. The soft tissue is situated in a central cavity of tooth and filled with connective tissue, vessels and nerves. This is the dental pulp. The hard tissue, like bone, consists of a densely mineralized organic matrix. The hard substance forms the wall and roof of the tooth and consists of three calcified tissues. Enamel surrounded the dentine and forms the major portion of the crown. Then dentine surrounds the pulp cavity, and then cementum surrounds the dentine in alveolus. Now we'll see first enamel. Origin of enamel is of ectodermal origin, whereas the other structure of the teeth are derived from mesoderm. The amoeboblast forms the enamel which covers only the crown of tooth. Chemical composition of enamel is due to its high content of mineral salts and their crystalline arrangement. Enamel is the hardest calcified tissues in the human body, forming a highly resistant covering or crown of the tooth and is whitish in color. Enamel is fundamentally similar to bone and dentine in composition, but contains less water and more inorganic matters. The enamel contains 96% inorganic solids, mainly calcium and phosphorus in a crystalline form, some keratin proteins, and the balance being water. Morphology of this enamel. The mature enamel is a completely compact, acellular, and avascular structure, organized in the form of elongated hexagonal rods or prisms. Each enamel prism constitutes the morphological unit of structures of enamel and is formed by one amoeboblast in its entirety. It is about 4 micron wide and the various enamel prisms are held together by minute amounts of interprismatic substance. The prisms and the intervening material between them are composed of crystallite, that is called hydroxyapatite crystals. That ground sections of the tooth shows striations due to the peculiar arrangement of these prisms and striae of ridges. These are imbrications or incremental lines known as striae of ridges are frequently observed in sections of enamel. They are attributed to the interment growth of enamel. These incremental lines originate at the junction of dentine and enamel and extend to the free surface of crown in an area paralleling the surface of the dentine. This strata appears concentrically arranged in transverse sections of the crown. Dentine, it is also called as ivory. The major part of the tooth is composed of dentine. Dentine is avascular and is devoid of cell bodies. Dentine supports enamel and acts as the skeleton of tooth. It surrounds the dental pulp and its pulpal surface is lined by one or several layers of cylindrical cells called odontoblasts. These odontoblasts are responsible for the manufacture of the dentine throughout the life of tooth. The dentine is an extremely dense calcified tissue similar to bone but harder than the bone because of its higher content of calcium salts. Chemically, it is composed of an organic part about 20%, which consists of collagenous fibers, Tome's dental fibrils, and cementing substance. The inorganic part is about 67% of dentin is present in a form of hydroxyapatite crystals. It also contains moisture of about 13%. The dentin constitute the bulk of a tooth and consist of calcified ground substance. Mature dentin is form of calcified component called apatite and an organic component of a fine collagenous fibril arranged in the form of a bundle, dential tubules or canaliculi. The outstanding morphological characteristics of dentin matrix is its system of dential tubules or canaliculi. These are 3 micron wide and are equally spaced and arranged in parallel rows. They enter from the pulp cavity to the periphery of a dentin in an S form, giving dentins a radially striated appearance. And third one is a Tome's dental fibers. In the living state, the dental tubules are penetrated by thin protoplasmic processes of odontoblasts, termed as fibrils of Tome or dental fibrils. The dentin is completely transversed by these processes, which are thus the vital component of a dentin and place the dentin in direct communications with the dental pulp. Third component is a cementum. It is calcified tissue that forms an extremely thin covering around the periphery of root. All the tooth components, cementum most clearly resembles a compact bone in origin, appearance and composition. It is made up of cells, collagen fibers, and a calcified ground substance. Cementogenesis is mesenchymal cells, also called as a cementoblast. They differentiate and enlarge to become fairly large round cells which lie adjacent to the root. Ground substance is elaborated. Fibers penetrate the ground substance extending to the surface of root and then mineralization occurs. The end result of this process is one in which a thin layer of calcified tissue is deposited on and firmly attached to the entire root surface. Here embedded in the cementum are the collagenous fibers of the periodontal membrane. These are typical Sharpie's fibers and are responsible for supporting the tooth in its socket or alveolus. Cementum is akin to bone and dentin in its chemical composition. It is composed of 70% inorganic mineral salts and approximately 30% of organic matters and moisture. Next tissue is dental pulp. It is basically a loose connective tissue confined within the pulp cavity. It originates from the dental papilla containing condensed mesenchymes, which gives rise to its cellular component. Fibers are non-existent or spare, but cellular elements are extremely plentiful. Topographically, the pulp cavity is divided into two areas: the pulp horn or horns, occupying the region roughly corresponding to the cusp, and the narrow slender portions occupying the space usually referred to as the root canal. Blood vessels pass into the pulp via the apical foramen of the root canal. The entire space occupied by the dental pulp is referred to the pulp cavity. The dental pulp is essential for nourishment and upkeep of dentin. The amount of pulp in the tooth dwindles with the age and additional dentin is produced. Cellularity of the pulp likewise decreases and the proportions of collagen fiber decreases in the course of time. Apart from this, there are some dental supporting tissues. Peridental ligaments or periodontal membrane. The periodontal membrane or the attachment apparatus is a special types of dense fibrous connective tissue lying in a narrow space between the cementum and the bony socket. It is similar to the periosteum of bone but is devoid of elastic fibers. There is an abundance of dense collagenous fibers arranged in coarse compact bundles, imparting it a characteristics of ligament. Some of the principal collagen fibers extend from the cementum to the alveolar wall via the periodontal membranes. The fibers are so arranged that the pressure exerted during mastication do not reach to the bone directly but are borne by them. Different cells such as osteoblast, cementoblast, and fibroblast are present in the periodontal membrane. A vitalizing plexus of small blood lymphatics vessels passes through the periodontal membrane, which also contains numerous nerve fibers that transmit sensation of touch, pressure, and heat. The main functions of periodontal membrane is to suspend the tooth and secure its position in the alveolus by attaching its root to the alveolar wall, gingival connective tissues, and roots of the other teeth. The gum or gingiva. It forms a collar of soft tissue around the neck of each tooth. It is modified part of a oral mucosa. The mucous membrane consists of non-keratinized stratified squamous epithelium that covers a lamina propria of dense connective tissue. The lamina propria is firmly bound to the periosteum of a jaw. It has tall connective tissue papille. The undersurface of the epithelium binds itself to the tooth enamel by means of its basement membrane and forms that so-called epithelial attachment of a Gottlieb. The outer surface of epithelium usually shows a slight degree of cornification in response to abrasions caused by coarse food particles. The gingiva differs from the oral mucosa proper in that it lacks a complete submucosa. Glands are also absent in the gum. The chief functions of this tissue is protection as it serves to prevent injury and infections to the deeper tissue. Another tissue is alveolus or socket. It is a bony crypt in which the tooth is suspended and is in immediate contact with the periodontal membrane. Histologically, the alveolus proper is composed of thin laminae of bone in which the collagen fibers are not arranged in a typical lamellar pattern seen in an adult bone. The distal end of the collagen fibers of periodontal membrane penetrate into the bone and cementum, forming a connecting bridge between these structures. Numerous trabecules of supporting bone between the alveolus and the external parts of jaw are advantageously disposed to take up the space that the teeth transmit to the bone surrounding them. The blood vessels and nerves run through the alveolar bone and the foramen of a root to enter the pulp.